Start the record. Thank you. So tonight we are starting this two-part series called, this one is on native seed collection. And on Saturday, we'll have a field class on native seed cleaning. Tonight's class is going to be given by Vanessa Robertson Rojas, who holds a master's degree in environmental science and has 15 years of experience in ecological restoration in the Pacific Northwest, with a focus on plant community dynamics. She leads the Understory Initiative's Rogue Basin plant material development and oversees restoration and monitoring across diverse habitats. Vanessa coordinates implementation with partners and leads plant material procurement and seeding activities. Vanessa coordinates the Southwest Oregon Pollinator Collaborative. And then on Saturday, the class that is paired with this will be a field class at the Understory Initiative facility in Ashland. And Catherine Prive will give that class. Catherine founded and serves as executive director of the Understory Initiative. She's a lead architect of multiple regional seed strategies and a co-author of the Oregon Native Seed Strategy. With more than 10 years of experience in ecological restoration and a master's degree in population genetics and ecological restoration from Morgan State University, she specializes in seed system design, grower coordination, and interagency collaboration. Catherine coordinates the Rogue Native Plant Partnership, including information sharing and public outreach events related to seed collection, cleaning, seeding, and planting. And with that, I am happy to turn this over to Vanessa. And I will stop my share. Thank you, Vanessa.
Okay, I'm just going to get my slideshow going. Thank you for that introduction, Marcy. How does that look for everybody? Good.
That's great.
As Marcy mentioned, I work for an organization in the Rogue Basin, or based out of the Rogue Basin, called the Understory Initiative. Stands to reason we work primarily on understory plants, and many of us are plant nerds in one capacity or another. Today, I'm going to talk about native seed collection for restoration projects. And I feel like this topic is really dynamic and dense and has a lot of different facets to it. And I probably won't cover everything tonight. So I'm going to talk for 45 minutes to an hour, and we'll have questions at the end. And we can sort of have a formal question and answer from the chat, or more of a discussion, whatever's clever. I think we have until 7:30, so we'll try and make sure we have time at the end for that. The Understory Initiative focuses on facilitating the restoration and conservation of native species through habitat through partnership and community engagement. Like I said, basically we work on the understory. We have a couple of different ways that we focus our work. We do partnership and outreach and sort of the restoration community collaborative work that a lot of Catherine's programs center. We do restoration, just prepping the landscape and restoring native plants to the landscape, and monitoring of those landscapes. And then we also have sort of a fourth part of the work we do that's sort of its own standalone place-based project and land-centered project, Vesper Meadow. We collaborate with them, but they really do have their own mission and set of missions and values that they enact on the landscape. We've been around since 2018. I joined in 2020. And today I'm going to talk to you about a big part of our work. Again, we'll try and stick with the program. I feel like I have a lot of divergences that I'm prone to, but I'll try and keep it as reasonable as I can. We'll start kind of big with why seeds even matter, how we use seeds or work with seeds in our restoration. And then I'm going to talk about a case study in the Lake Creek area. That's a project we've been working on for a few years. And within that project, I'm really going to walk you all through the multi-step seed increase process, and then talk about some of the things we've learned in that work and more broadly in TUI's work. And at the end, there will be a list of some resources that I mentioned during the talk that I can share afterwards if people would like to have access to that. So wild collected seeds are really important, first and foremost, I think, for the genetic diversity that they bring to the landscape. In the Klamath-Siskiyou region, our native plants have really been through a lot. They've been through a long era of having unique soil influences, an escaped glaciation history that not all plants in North America have experienced. And they're really resilient because of this. And many species that are globally common, like yarrow, for example, might have a unique set of genetic inheritance because of this experience on the landscape here. When we think about climate change, extreme weather events, human activities, that resilience can be uniquely beneficial to restoration in our lives in general. Wild collections are used for seed increase to really try and capture the breadth of that resilience. And then they're used to create many pounds of seeds that go back out on the landscape. Long-term conservation that is plant-based is globally valuable. You know, when you establish populations of native plants back on the landscape after wildfire, for example, you reduce the risk of that same wildfire happening in the same intensity, high intensity in the future. Native plants, there's a lot of literature about native plants really being effective at carbon storage and offering pollinator habitat that impact, you know, agricultural practices and wildlife forage. So within the restoration context, we really, as plant people, benefit from thinking about making a plan, implementing the steps we've described, observing it, recognizing places where our plan did not align with what we saw on the landscape, and adaptively managing a new plan or new activities in the future. In our work, we do this somewhat annually, and it kind of matches the annual lifestyle of plants. We might implement some actions in, you know, a given growing season, observe the plant response, and then make a new plan for the following growing season. This is a really common concept used to describe our management and restoration context today. Historically, people had more of a top-down approach of just managing acres, putting plants on the ground, walking away, and treating new acres. So for today, I think we're going to spend a lot of this time talking about the UROI project, which is the Upper Rogue Oak Initiative that on the right side of my screen, or where my cursor is based in the Butte Falls, Lake Creek, Eagle Point area. That has this initiative is a project put forth by the Klamath-Siskiyou Oak Network, or KSON. And KSON is a partnership that works with private landowners, public land managers, local restoration practitioners, and other conservation-based practitioners. It's a large partnership that is based on restoring oak systems specifically in our area. The Butte Falls area, where the Upper Rogue Oak Initiative, or I'm just going to refer to it as the UROI project from here forward, is mostly about 2,000 feet, which is markedly different from a lot of the work that happens in our restoration world in Medford, for example, that is maybe lower to mid elevation. The UROI project is over 3,000 acres in the broader footprint of it. The seeding footprint within that 3,000 acre project is around 1,000 acres. We don't seed every inch of every 1,000 acres, but our target is to disperse the most appropriate plant materials across as much of that 1,000 acres as we can. Our plant materials include a partial shade mix and a full sun mix at this time. However, future work through the Klamath Oak Network could support additional specialized plant materials. So the UROI goals overall are to decrease white and black oak encroachment by conifers. Additionally, we recognize that a side effect of reducing conifer encroachment is fuels reduction. It's not the primary goal, but it is sort of a positive benefit to the work. This project has a nice 6-year timeline from 2023 to 2028. So TUI started working with this group, I think, in 2021, and the UROI project was funded through the Caisson Partnership in 2023 through OWEB and the NIFWIF Foundation. It isn't common for restoration projects of this scale to have such a long implementation and seeding and monitoring period, but it is becoming more the norm as our work evolves, as we recognize that planet Earth really takes time to recover from the initial degraded conditions and the restoration actions. TUI recognized that our specific goals include some site preparation and primarily providing as much genetically and ecologically appropriate plant material as possible. And we also conduct pre and post seeding vegetation monitoring. The monitoring that we have conducted so far seeks to characterize the site before the encroachment thinning is done, and then we will go back and characterize the site after. We also are developing some more in-depth plot networks within the UROI footprint to more accurately observe and monitor seedling emergence in some of our targeted seeding areas that may not fall into the broader plot network of the UROI project. So for creating seeds, this process is really complex and it's iterative. We revise often. And I think it's really important for us to keep an eye on our ability to revise and be adaptive. When we are working with plant materials, I feel it's really obvious really quickly that you are sort of at the whims of planet Earth, and you can't always hit your targets on the year you meant to hit them. So we are always revising our wild collection plans, our seed increase farming, and our seeding activities. But our initial goals are always to do a fair bit of research on the front end. We do site characterization with monitoring, like I mentioned. We use web resources to create an exhaustive species list, and then we refine that species list with a variety of tools and processes. From that research process, we wild collect seeds, put them into bed production, and once those beds yield crops, we manage our inventory internally so that we can create and mix our seed mixes and apply them to the landscape. And on the left side of my screen, we basically go from these teeny little bags of all this fluffy plant material coming off the landscape that is hand collected by TUI staff and volunteers into these beautiful seed mixes that have a pretty nice diversity of species. So I'm going to walk you through all of these steps, and I'm going to still try and keep us on track for time. Okay, so for site characterization, we do some formal monitoring. You can do informal monitoring. You can walk through a site and create a species list, or take some photos of plants. You can walk with a botanist and learn new plants. We do monitoring because it's a part of our wheelhouse to provide ecological data to our partners. This is a way that we show that we actually made a change in the landscape. It's not the only way, however, to characterize your site. What I have on the left side of my screen is a transect with a couple of squares in it. This is one way that we might develop a series of 1-meter plots, in this case, that we laid out systematically across a transect in order to collect data in a 1-meter square which you are seeing on the right side looking down at the ground. These are obviously from different sites, but it's the same protocol that we commonly use. You look down at the ground, you count all the different species that are there and associate each species with a percent cover value. And then you look at it on a computer and do all kinds of analyses to understand the community that is present. Some interesting things that can come out of that analysis is just, general summaries where we look at each species and how common they were. What we often see in our pre-data collection or our pre-project data is a high cover of exotic plants. Plants that aren't from here, that have been introduced onto the landscape, that might be creating some kind of a monoculture that we would like to remove from the landscape and replace with a diversity of native plants. So that's what you're seeing on the left here is sort of a summary table of our species list here at the top. And then in the bottom left, there is just a series of plots or sections followed by a column of each section's total exotic cover, its native cover, and additional ground cover. On the right side of the screen, we split the species present and percent cover data into a couple of different groups that sort of match the types of forest cover you would see on the landscape. This is a characterizing analysis that really helps us divide the landscape into a couple of different plant associations. Again, none of this is required, but it's some fun stuff that we do that I think we will come out with some formal reports about in the next few years if you are interested. What results from this is an interesting list of plants that associate with each other under different forest cover types. So you can see conifer cover over here, and you might associate this group with a more open canopy conifer cover forest type. On the other side of this visual, you would see closed canopy oak cover, where there's maybe a lot of oaks that close the canopy, and these species are found in the understory. So we do this work where we collect data on site, and then we compare it sometimes to the Oregon flora species list for different seed zones or different sites. This might give us an idea of other plants that could or historically have occurred on the landscape that are maybe occurring in trace quantities that were not detected in our plots. It's important to think about this through a historic lens and through a trace species lens, because what's present on the landscape might not always be what you want to restore to. You might want to restore some extant species back onto the site, knowing that they used to be there and they've been removed or overcome by exotic plants. So we review our community data, our pre-existing species list, and then I usually take a peek on iNaturalist from near the project area. Sometimes people show a lot of records of things that gives me a sense of how frequently some of these native plants are here and are not showing up in our plot networks. And then we combine all of these things to create a really exhaustive list. Sorry. We want to start with, a really broad look at the plants that might be occurring on the landscape and are occurring on the landscape. And then we refine. I usually start with splitting the exhaustive species list into a couple of key groups. One of those groups is, we call them workhorse species. And I think this is a little bit of an extractive way to talk about plants, and we might be sort of revising this language in the future. What we mean by workhorse species is species that are really common, established readily, and provide a lot of ecological lift on site. For grasses, it's species that we're really familiar with. If folks have been out on the landscape looking for bunch grasses, you may have come across the Lemon's needlegrass, our brome species. We have some lovely native bromes. The festucas that occur across conifer, savanna, and chaparral habitats, and then some of our meadow species like Coleria macrantha and Vulpia microstachys. These species are really common. They're seen everywhere. When we do our surveys, they usually show up in our data. And they also really do well in farming um, seed production contexts, which is an important part of our refinement process. Some other workhorse species or generalist species that we include in our mixes are Oregon sunshine, Prunella vulgaris, yarrow, Clarkia purpurea, or the wine cup that you may be familiar with, and Gilia capitata. These plants are, again, common on the landscape and fairly easy to establish in seed production beds. And they do well in our mixes. These species thus occupy a pretty large component of each seed mix that we use them for. Then we go back and think about plants we've seen on the landscape and plants that we have seen in our species list that might not be well represented in our data, but we recognize that they have ecological importance. They might be really difficult to farm. They might not even be farmable in the timeline that we're talking about. And they might not be something that was easy to locate to make a wild collection from. When we identify these trace species as ecologically important or culturally important, we do kind of go out of our way to make sure they become a part of the mix. On the far left, you can see the Penstemon dustis that is pretty hard to farm, but we did some in-house farming at the OSU Extension actually for a few years to get a couple pounds of this into our 4,000 pound inventory. And the same thing with the Horkelia and the Hemizonia, the alliums. For the allium amplectans that we are looking to include in this UROI project, we haven't really been able to farm it or find the right situation to produce seed increase of it. So we have decided to just collect sparsely on the landscape. That's why it says WC in parentheses under it, because it is only something we wild collect. And we will, reintroduce those small trace quantities back into meadow systems in targeted seeding applications. Waiithia angustifolia, we do actually farm this one as well as some of the others. It just doesn't produce oodles and oodles of seeds, but it's pretty important. I didn't say this when I started, but I should have. We use a lot of our own in-house imagery just to really document our plants in situ in the rogue. But when I don't use my own imagery, I do try and credit Oregon Flora or whoever they are taken by. Once we worked with our exhaustive list to include our workhorse species, our trace species, within the UROI project team, we did hear back from some of our Indigenous partners that they would have liked to have seen culturally important plants occupy a bigger component of the mix, primarily the meadow mix. And one of the big themes that we heard was that bulb species were really important in this area and would have likely been here. So we worked to increase the amount of camas we included and calochortus, as you may be familiar with the hairy cat's ear lily and Perideridia gardneri. We weren't originally too sure that we would be able to really do seed production of these species, but we're making an earnest effort at it. And the camas seed production is going pretty well. The calochortus we've just put in this year, and we just got some, good germination and establishment specs on Perideridia this winter, so we might be able to do some of that soon. We wouldn't have been able to do that without the input of our Indigenous partners highlighting how important it is to include that into the mix. So the Indigenous lens can be a really important part of our planning process. The pollinator resources filter is one thing we go back and use to refine our seed mix composition. You'll see here that the current mix that we use spans a floral period from March to November of a given year. We do want to make sure that we're offering pollinator resources for as wide of the floral calendar year as we can, the biggest proportion of that year, just because we recognize that pollinators in our region are so imperiled and that under climate change conditions, their resources are becoming sparser and sparser. It would probably be much easier and much cheaper to include fewer species that mostly bloom in June. I just, from looking back on all this work, we add a lot of complexity into our mixes to really try and incorporate ecological complexity. And I think we're achieving that. It's just a fair bit of extra work. And this is a pretty reduced list, right? This is maybe 10 to 15 species. For the URI project, we do have 30 to 40 species that we are including in our plant material list. So you've gone out and seen your site, you've made an exhaustive list, you've refined it a little bit, you've put it through some filters, and now you're ready to start scouting. And I think this is a really fun part of the project. You go out and you see the landscape and learn about um, where these plants might be occurring, and you can make some notes in your journal or your phone or your data collection application that maybe you're using to document where and when you saw the plants you are looking for. Um, we refer to Oregon Flora to check out, some of the historic sightings of a plant, and we might go visit a site based on that. This image in sort of the center of the screen is an Oregon Flora map of where Bromus sitchensis var. Maritimus has occurred on the Oregon coast. It's a great example of a very sparsely documented plant because of the new taxonomy in Jepson and the Oregon Flora taxa. This plant seems somewhat unrepresented. However, when you go out onto the South Coast, turns out Bromus maritimus basically is everywhere. It's just not recorded as Bromus sitchensis var. Maritimus. So it's important to go out and double check those records. And then we look at INAT and we see something similar here for the seaside brome under Bromus maritimus. And that tells me that not only is there a little confusion about the name of this plant, it is not being documented in the wild. I should have put another map showing our collection records of it. It's somewhat common in coastal meadows. It's just a good example of comparing some internet resources with real life and going out and scouting and checking and searching high and low for your population. So you've done your species collection. You've looked at online plant records. Maybe you've checked a couple of different resources. And it seems reasonable maybe to get out into the world in spring and summer to do some scouting. At our organization, we do a lot of data collection around the scouting material or scouting information. We go out and collect, a lat and a long, the phenologic phase of the species, the number of individuals in a population. We also associate the location with an ecoregion that gives us sort of like a seed zone level bit of information. We record land ownership and all kinds of information. And once we come back from scouting, we might review, how much we found and where were the best populations in order to start doing our permit applications. Then we plan our summer and fall collections based on the phenologic information we gathered in the field. And that just means that we look at, how much of the population was flowering at that time, and we make an educated guess about when those flowers will ripen into seeds. And then we get to cleaning once we've made those collections. Here's a list, a more specific list of all of the information we collect. Again, I can share this presentation with folks if you're looking for some of this information. I can't imagine a casual seed collector would be looking for like the seed zone plus the region and like a formal latitude and longitude, but if you want it, it's available. We can share that info of what all we collect. One thing I think that's really interesting about this work is that estimation of ripening. Sometimes I go on the CalFlora website to review the bloom window that has been recorded, and CalFlora has a great little calendar. You can see this on the right side of the screen where it shows the months and when this plant has been documented to be blooming. From this, you can kind of deduce a ripening period. It's doesn't seem really possible to have any kind of highly accurate predictive model about when a plant will ripen. It is based on aspect, slope, elevation, climate, and it changes interannually. I think of this species in this picture, Plectridis congesta, as a pretty reliable, we're going to collect it in late May, early June species. But this graph over here shows that it blooms from March until June, which is just an expression of its, geographic diversity or plasticity. We then, make this guess and go check in person. You can test a seed's readiness by hand rolling it and feeling if it's hard in between your fingertips. You can cut a seed open and see if it's still moist inside or has it truly dried and is it ready for collection. That moisture check on the outside of the plant also can be helpful. You can also do a touch test for how easily that plant the seed comes off the plant. We collect using paper bags or something that allows the plant to continue to dry once it's been collected. I don't recommend using like a plastic Ziploc because they'll kind of just rot inside, even if they're seemingly completely dry. It's always surprising that there's still some stuff in there that could rot. We also put our collections in a shady, dry, but pretty warm place. So not your fridge, maybe in your carport or maybe in a dark room in your house that gets, good airflow. This allows the plant to just keep curing, which I think is an important part of seed physiology that maybe is sometimes overlooked, is that many plants in the wild would have a curing process where they fall to the ground and continue to just get kind of weathered by the sun and the heat and whatever. So once we've gone through a lot of these processes to like clarify everything and research everything and scout everything, and maybe we're starting to make collections, I usually have some kind of list like this where I have the species, the 6-letter code, a target quantity that I would need in order to set up a seed increase bed, and then I have estimated a ripening time. And a monthly is about as precise as I choose to get because it just varies so much. I list the best locations to collect species from, and then I usually assign like a priority of importance to it. And then I'll split things out by my permitting needs, given the land ownership type that was documented during the scouting phase. And from this, we've gotten some cool information over the years. We worked with a data analyst a few years ago to visually represent whether or not the phenology is changing in our collections, and the ripening dates do seem like they're moving a little bit. They're not staying the same interannually, which is interesting. I guess this is a little bit of redundant information, but we associate some of the phenology with these other pieces of information to really document all of the encounters that we are seeing on the landscape. So then we're out here, we're making our collections. You've gone out and made your species list and checked if it was even there, and you waited patiently for the plants to ripen. This species example is Perideridia gardneri. That is an important plant for the UROI project, but we don't have it in production yet. So we do a significant wild collection every year. And this picture on the left is of it in bloom. It then ripens into these beautiful seeds on the sort of rounded umbel shape that is common of the carrot family that it's a part of. And here we are out on the landscape collecting with our caisson partners who happily enjoy some field work collecting seeds. It's pretty nice work collecting seeds. Here you can see, like I mentioned, we're doing it by hand. Um, and we do what's called like a seed stripping motion, where you wrap your hand around the inflorescence and maybe pull upwards and away or and with a twisting motion. This is what I've found to be the most like bang for your buck for every time you're bending over. I am not 21 anymore, so I do try and have some like field ergonomics. Some interesting considerations to make while making seed collections. You will see the genetic breadth and depth of our region here. Plants have all of these really special strategies and mechanisms for dispersing seeds, for protecting their seeds, for enriching their seeds. And it really is just a diverse ballgame. There is no straightforward method for really handling all species. So we make a lot of um, in situ decisions. On the left side of the screen here, we have coastal iris that I thought was really interesting. We really showed up um, on time to make this collection. However, the seeds inside of the pod were ripe, but the pod was so juicy and wet still. It was um, kind of shocking to see how like disparate the levels of dryness could be between the seed materials themselves and the pod that was like adjacent to it. They were starting to open. We could see that the seeds were ripe, so we made the collection. And in letting them just dry over a screen for a 2-day period, they popped open, just flung seeds everywhere. And I had put a little plastic cap over it to prevent losing the seeds. But they also just dried out like paper thin within a couple of days. It was really amazing how quickly those plants digest, I guess is the word for it. These seeds are from a forest adjacent to these sand dunes that are kind of a great example of the need for sun protection in the work that we do. In the center image, there are two phacelias right next to each other coming out of the same basal rosette, and one of them is completely ripe and one of them is not. And this happens all the time. Plants do this in the wild in order to cover a broader window of time to put their little precious babies out onto the landscape. What we do in response to this is only take the ripe stuff in most cases. We usually collect only 20% of any given population that must be over 50 individuals in order to even be considered for collection. By having those standards, we minimize negative impacts to plants over long periods of time. So we let those indeterminate seeds ripen on their own later and have their lives out in the world. It is important for us to recognize that in these instances, we are creating a selection event. And so we don't want to repeat that every single time. If this indeterminate collection happened on June 6th of a given year, I might not go out in June to get Phacelia the following year of the same species. Mattia elegans on the right here is a really interesting species that's also crazy indeterminate, but it's not clustered the way this Phacelia is. And when you touch it, it just seems to scatter. So you might get 5 plants on a seed if you were trying to do the hand stripping motion. What we do instead for that one is use a flat cardboard box and just tap into it. This is not some brand new invention. Indigenous people collected Mattia elegans and Mattia sativa in particular this way since time immemorial. So we again can learn from our Indigenous partners about how to interact with these plants. Grass in the field. When you're doing wild collection of grasses, I feel like it is a lesson in training your eye. On the left here, we have these tiny little Festuca romeri seed heads that you use to identify the plant. The second image over is what it looks like in the wild. When you are looking at it in a production bed, like this third image here, it's so easy to know. This is Poa secunda, by the way. It's not the festuca. It's easy to know that you have this one species. But I find the mixed meadow that is high quality grass habitat has a lot of natives on the landscape. They all kind of co-occur and they mix and they mingle and create the complexity that we love on our landscape. And it makes seed collecting, tricky, I guess. On the far right, I have a picture again of Ignathra mammonii, the lemon's needlegrass that has these really beautiful awns. You can see that sort of change when the plant ripens and they get a little bent in a certain sort of shape that you can start to see on the landscape as you hone in on that species while doing your collections. For Forbes species, again, we make all of these sort of special considerations for how we are going to collect this plant. If you can imagine walking out onto the landscape and seeing all these poofy little dandelion looking sort of species, the Acuricina or the blowwives, it's difficult to collect without knocking your collection onto the ground instead of into your bag. So we spend some time sometimes just crawling around on our hands and knees, plucking precious little tufts of seeds into our bags. Trichostemma linatum is the vinegar weed that you may have smelled in November or October of a given year. It is usually not ripe until really late in the season. It is an incredible plant for late season pollinator habitat. It's one of the few plants really blooming that late into the season while those pollinators are experiencing a ton of drought and a lack of resources. So we wait until October and November and go look for the vinegar weed, which is usually pretty easy to find because it stinks to high heaven and it's one of the few remaining plants out there. This picture I just love of the tritornatum. I was looking for it for years and then just happened to stumble upon 5 acres of it. And sometimes that's just the way it goes. You scout and you scout, and while you're walking somewhere else, you find the most incredible population that nobody's ever put in INAT or anything. And so you wouldn't really find it unless you were walking just through a, part of our landscape. One interesting forb species that we experienced in the Uroy footprint was Clarkia gracilis that you can see in the center here. We typically assume that the Clarkias of interest are Clarkia purpurea and Clarkia rhomboidea. Once we were working in the Uroy landscape and I had already put our Clarkia species into production, we learned Clarkia gracilis is everywhere. It's super common. It spans a couple of habitat types, and it's just something we would consider including in the future. So now you've got your collections and you've let them dry, and it's these, you have all your little bags of fluff. When it's a small collection, it really makes sense to do some of these low-tech hand cleaning methods. On the left side here, there is a series of screens that have different mesh sizes that allow different sizes of plant material through. And what you can see is that the seed stays at the top if you select a mesh size that's too small to let your seed pass through, but all the dust comes out the bottom. Sometimes the dust is the same size as the seed, so you might want to wind winnow by flipping it in a flat surface and in a gentle breeze or blowing on it or in front of a fan. Something kind of like this, where you could just pour it out, and you can see here at the bottom that after the fan has had its way with the fluffy bits, they're separated and they blew further because they don't have a heavy little seed embryo in them weighing them down like the seeds do over here. Sometimes we clean with our friends because it's tedious, redundant, hot, sticky, mask requiring work. We worked with some kids over here to really thresh a buckwheat, a coastal buckwheat. And that was really fun. It was a great partnership because those kids loved ripping those plants apart. And it really saved me a lot of rigmarole. And here we are using our air machine. That is a sort of sophisticated way of doing that agitating and blowing workflow with a bunch of our pals to clean some seeds. And then once we've cleaned everything and we've weighed it, we've labeled our bags, we put all of our seed information into a database that our ED Catherine has created um, in Baserow, which is an open source software. We use this to manage the wild collections coming in, the collections we give to our farmers, the crops that are produced with those wild collections, and eventually the seed mixes that we will pull from those crops. As I mentioned, we take our wild collections and turn them into these grand seed beds, and it is usually a sight to behold. They are just, you don't see plants in this quantity in the wild, and it's always kind of a beautiful thing to see. Seed farming is really difficult. Um, our farmers work really hard. These plants have not been bred to adhere to any sort of easy workflow. In very few cases are they just, you cut it, you clean it, and it's just a breeze. One benefit to native seed farming is that the nutrient and like feeding needs of these plants are often much lower because they are plants that are used to living in very depauperate soils with no fertilizer. And these plants can be somewhat low water needs. I wouldn't say that any of our plants I really recommend just not watering at all. Uh, you can, though, drop the water off at a time of the year to really instigate ripening or help the plant mature on a timeline, say, before summer ends. Like I mentioned, it's just really beautiful to see these huge beds of all these beautiful plants. The way that these plants work is really variable, even in seed production. In the center image, you can see some of the harvesting has happened by just cutting the heads off and putting it into a container. With our showy milkweed on the left here, we hand harvest the pods, let them pop open, and sometimes we clean the fluff using a tumbler. Sometimes we burn the fluff off using a lighter. Yarrow uses an air machine and is usually harvested with some kind of singular cut or a combine. So for the URI project, we started our production design in 2022. We mapped our populations and started our permitting and started our wild collections. And then we spent, another year just really working on rounding out the collections and revising our production plan. Because we worked with the U.S. Fish and Wildlife Service in 2021 and 22 to get this process started, we had like a uniquely robust foundation inventory to choose from, which is not common in all of our projects. Again, the Caisson team is just like a fabulous partnership that has allowed us to do a really good job on our plant material work. So after 2022 and 2023 go by, we've made our collections and have cleaned and inventoried everything. We start putting plants in production with farmers, and then it takes several years for them to yield. In 2025, we started finally getting our really big crops back from farmers. Um, it is a magical thing to have a pallet of like 800 pounds of grass seed dropped off to your seed facility. It's, was 4 years in the making, and the results are really worth all of the work that we put into upfront. Okay, so now we have our seeds and we start thinking about our seeding application around 2024-25. As we know, these crops are starting to come in. Within the URI acreage, the Lomakatsi Restoration Project has done the thinning work and burned the piles that create bare ground for us to seed into. And we start developing and mixing our partial shade mix. In 2025, we put 85 pounds of this out onto the landscape. It had 28 species in it. The majority of them are sourced from the Lake Creek area. It is 35% grass, which is unusual. Most seed mixes have 60 to 70% grass because grass does really well in seed production. And we designed this mix to occur between 30 and 60% shade. We also developed our full sun mix. There weren't as many piles burned in the full sun habitat type in the URA project, so we chose to seed only 15 pounds of that on the landscape. We are gearing up for a much bigger meadow seeding after a large prescribed fire meadow burn in either spring or fall of this year. This mix has 24 species. They're also sourced from Lake Creek and is 45% graminoid. And this mix is meant to be applied in 0% shade up to 30% shade. And I just want to talk about the site that these are going on. We realized, and you may be able to see this from this aerial imagery, that across our site, we had some places like this seeding area 5 here that has really high canopy cover. In seeding area 2 is a bit of a mix. And then we see seeding area 1 has much more acreage of this full sun habitat type. And we went through and did a little bit of acreage math to try and deduce approximately how much of each acreage we needed to pull seeds for from our inventory. This is, again, a thing that we kind of iterate in order to make sure we have the right mix for the right amount of acres, but you can't always know in advance exactly how many full sun acres are going to be created by the cutting crew, because they go through and do their work based on the forestry prescription, and you don't know what's out there on the landscape until you start doing your cutting. Another interesting part of the mix pulling and distribution is that there's a lower seeding rate for the forest mix versus the meadow mix or the full sun mix, because you just have less resources around for plants to establish, so you don't need to put as many individual seeds on the landscape. So here's just a pie chart kind of showing what our mix is composed of. This is our entire production plan, actually, of all the species we grow. And it doesn't have everything in it, but it has everything that's over like 0.5%. I guess it doesn't include the total trace species. But you can see that there's a pretty good distribution of species across the mix. It's not just 50 pounds of Elamis glaucus and 5 pounds of everything else. We do try and do a good job of really diversifying our plant materials. Okay, so to recap, the seeding in the forest is burn pile seeding. We apply seeds on the landscape in late September into December in order to give the plants moisture and cold stratification in situ before the next spring. We do not recommend seeding outside of this window for any plant materials that we have anything to do with. In this section here, we did mostly the partial shade mix. All of these blue dots are little burn pile scars that we documented. At each of these plots, we took spatial information, seeding rate, ground cover, seed mix, and this type of seed mix that was applied to the burn pile. And we will be going back this spring to monitor a subset of these plots. What you are documenting when you go back to measure a seeding within like a year afterwards is called seedling emergence. And then we will go back in several years and see the seedling establishment that has happened maybe over 3 years. And here's just some pictures from seeding, and here's what it looks like really. It's a big scar on the ground, and there's a person seeding it and documenting it across the landscape. We do a little scuffing to really increase seed contact and decrease bird predation. What I don't have in my hand is a measuring cup. We usually do some precise calculations for how much seed needs to go in each burn pile. I think I have it in my bag there, but that's the general workflow for the seeding application. So once this is all said and done and we look back on things, Again, we review some of our lessons learned, some of the quantities and our spending efficacy, and there are oodles of lessons learned, but I'm going to keep it in the fun realm of like fun things that we can all use as people out in the world. I do think it's really important to remain open minded to unexpected species like that extra Clarkia. Clarkias are wonderful. They're just such a great little plant that offers so much habitat to pollinators. For there to be a third Clarkia should be good news, and we should adapt to that in the future. One thing we've dealt with that's super fun is how do you mix 100 pounds of seed, 1,000 pounds of seed? One of our targets is, of course, to make sure we seed 1,000 acres. But if you've hiked around the Siskiyous, packing out 4,000 pounds of seeds to distribute across 4,000 acres and mixing it all is just kind of a logistical ball of yarn, I would say, or whatever that phrase is. We also are learning about which species show up on the ground, like I mentioned with that plot network. And we've seen some species sort of unexpectedly really occupy space in the first year. The Gilia capitata in year one emergence is really high. And it's one of those high pollinator quality plants. You see beetles and butterflies all over it while it's blooming, and then it kind of dwindles after a disturbance. And it's one of those species that we've just been able to really embrace its early establishment on the landscape and then know that it'll fade out over time. And while it's doing that, it's providing tons of habitat in the process. What are the best seed ratios? Seed math is crazy. We do this math all over creation to try and create, the right proportions of percentages of the mix with consideration to the seed size. And here the seeds we're comparing are Asclepias speciosa, the showy milkweed, and yarrow seeds. And I would argue that there are exponentially more yarrow seeds in this situation because for one little pinch of yarrow, there are hundreds more seeds than there are of the Asclepias. And they both establish differently on the landscape. So the way that we plan to put these seeds out can really vary by seed size, emergence type, species and how they relate to each other. And I think that embracing all of that complexity is my really big take home message for the URI project and seeds in general. From this talk, I mentioned a bunch of resources and I have included links to them in this slide here. And I do want to point out that in iNaturalist, you just have to click on the Explore function to start looking around your area or your site for records. One thing that's really helpful and really cool that we use a lot or refer to a lot is the NRCS production manual. This manual was written for the Willamette Valley, and we are working on sort of a Rogue Valley version of this that might come out in 2027 that focuses on, seed production specs and seeding application guidance. This amount of information that this guide provides is no small endeavor. There are, more than 50 species in it, and they all have, the wild collection, ease of establishment, harvest, seed cleaning, everything for tons of species. But we need the same thing for the Rogue Basin because our region has such variable plant strategies, lifestyles, and genetic histories. Okay, I think we can have some questions now. Thanks for listening to me talk. I'm going to stop sharing my screen.
Okay, Vanessa, thank you very much. It's really interesting. I'm just looking at the chat. Artie asked, why are conifers able to outcompete native oaks?
Conifers begin to outcompete native oaks in the absence of fire, and I would argue humans tending the landscape. Historically, people really worked throughout the landscape to clear up areas around and under oaks so that they could maximize acorn growing or acorn production. People used fire to do this, and oaks are adapted to survive fire as seedlings better than conifers. Um, I wouldn't say that they completely outcompete them and kill them, though. What we have seen in this project is that conifers have a growth habit to grow straight up and become really tall, and then they shade out the oaks. And so, what this work really focuses on is removing conifers around oaks and allowing them to flourish again because they've just been hanging out in the shade, sort of waiting for some kind of stand replacing fire that would have removed conifers. Hi, Artie. Artie is one of the fabulous people who comes to help clean seeds with us every year, and it's always lovely to see you. Hi.
That's great. Thank you. And Jenny asked a question that actually I saw that you kind of answered. She asked, do you ever use Danthonia californica? It seems quite common in the Upper Rogue project area. So I had seen her question and I noticed it on your pie chart listed at 2.9%. Is there anything else you want to say about that?
I have so many things to say about Dan Cal. It was one of the earlier on in my presentation. It's also one of the like I would argue sort of I don't know like cornerstones of some of the open oak savanna habitat that is sort of the target for this project is to create more open oak habitat, but not everything full sun and not everything full shade, but some kind of matrix that has a heterogeneous canopy cover. And Danthonia is everywhere. You're totally right that in the Upper Rogue, especially in the Lake Creek area, these meadows have just oodles of it. And we think about this a lot. We saw that in that analysis and thought, oh, we better get some beds in production. So we made these wild collections because it was abundant on the landscape, sent it to our farmer, and he goes, great. I'll see you with a crop in like 3 or 4 years. And I'm looking at my timeline, like, oh boy, this is not great timing. And they don't produce very much. The farm beds are difficult to establish. The yield is low. It's very expensive. And when you see them on the landscape, they don't take very quickly. They don't take very well, and they're already there, which is an interesting element of this is this is why I don't base all of our production on what's already there. Because if you create a restoration action that disturbs the landscape, you kind of have some native plants that have been hanging on and can serve as sort of like an aid to the recovery of what you put out. And DanCal is everywhere, but it's hard to put more of it out. So is it important for us to spend, say, $8,000 a year on 50 pounds of it? That's how expensive it has been to create. That's kind of crazy. So we weigh these things and we've started, getting yields off of our beds. And I think that's another lesson learned is which crops are so expensive that it isn't really a benefit for us to grow it, especially if they're doing well on the landscape. I love that question. Think about it all the time.
Okay. So Gary Cleaver asked or made a comment. We happen to know is a seed farmer and a land steward. And when you mentioned that seed farmers work very hard, he said, "Yup, lovely and hard."
I know we worked with Gary. We worked with Gary for so long. I'm glad you could tune in, Gary. Hello as well. Nice to see you. Gary is somebody who works very hard farming seeds in Talent.
Yes, and he will be with us on Saturday, that the event. John Price asked, do you try to time seeding ideally before a rain?
This is, yeah, typically, yes. I think that's the general thought. Lately, I have been thinking that it might behoove us to kick it back a little later after a rain. When seeds are sitting on the ground in these burn piles, they're these light colored little dots on a black burn scar. And to a bird and all the birdie friends, it's a pretty easy snack. And I feel like when it starts to rain, the soot and charcoal in that burn pile is more easily like spread over the seed in such a way that the birds aren't going to eat it. I know birds will continue to ash bath in the burn scars throughout fall and winter, on dry days, but it seems like it decreases predation to just wait after a few weeks after the first rain. That's really persnickety. If people don't have that level of like flexibility in the seeding, many restoration practitioners start like September 15 and they just go until the seeding window closes.
Okay. And Caitlin asked, do you consider human aesthetic values when creating a seed mix, or just focus on how effectively the mix will create pollinator habitat?
It depends. We used to do this private land program where we were essentially giving technical assistance on how to create the habitat, but the landowner implemented it. And then we would provide them with seed materials for free. And in that case, people did a lot of cool things that really satisfied the human aesthetic. And sometimes people would ask us for a little more of the lupin or a little less of the mattia because it gets a little crazy in November. And all of that is just fine, I think. Within the Quezon project area, the requests for like human consideration that we have heard is from our indigenous partners. And that part of it is also fine to incorporate. It's great. I don't think it's always the case in wildlands where people have a lot of opinions because the amount of acres that you are creating is kind of the focus. It's continuous, it's large, you're looking for a high native take on the landscape. But I'm never opposed to it, I guess. I just feel like we as people are meant to live with the plants, and we, of course, care what it looks like.
Okay, and it concludes the questions people have submitted in the chat so far. So this was
Great.
really interesting. And I know you had mentioned when we spoke before this started that Catherine Prive was going to be here. I see that she is. And Catherine, do you want to say something about the class on that you will lead on Saturday?
Hi, everybody. I am making dinner for my daughter, so sorry I'm not more, I've been listening. But yeah, on Saturday, we're going to be doing some of the hands-on work of seed cleaning. You're going to get a taste of some of these species that Vanessa was so aptly talking about today. And it is very laid back and you don't need any experience. And we'll just get to know these seeds on a different level. And it's really helpful to our process because by having this help with the cleaning process and getting things ready, it lowers our costs, it lowers our effort, and many hands make quick work. So that would be great to see you all, and it'll be fun. And I have an espresso machine and I will make you a coffee if you come. So consider that.
Okay, thank you. We will look forward to it. I see another couple of questions have just been posted. Um, Miriam asked, have you tested using nursery techniques for increasing seed germination? For example, cover with grit, plant moist seeds before they go into dormancy, decreases 2-year germination period for some native seeds.
Yeah. One of the resources I listed was the Ranger Network, the RNGR network that lists a lot of these. When they are available, we absolutely refer to them for the propagation specs. I typically work with each farmer on the species that they have to make sure they have as much of the available specs as possible. And a lot of the cold strat for like the workhorse species is well documented. It might be a little different in our region, and it might vary by elevation or seed quality based on how well the wild collection was made. But a lot of that information is like the first thing we go to. And then when we don't have it, we've been doing some of our own in-house termination trialing to try and figure it out. And we figured some things out, and then some things is TBD. There are so many species in our region that have this really interesting strategy for being on the landscape that they might have a harder code to crack. But there are just so many species to figure out. So yeah, yes, I guess we do.
Great. Okay. And then Jenny said, hopefully Vanessa will come back and give us a talk in a couple of years when she can share which species or mixes worked well on the landscape.
I would hope so. That's the goal with what I'm calling like our seedling emergence data collection. It's sort of a new thing for us because we spent 5 years learning how to farm these seeds and wild collect all over the basin. And now the restoration community is really creating the bare ground that you would want to put these plants in. And the time is now to really start looking at the emergence and the establishment. And I imagine in 27 through 29 for the URI project, we will have some information, which will be great.
Please be sure to keep us in mind, and
Sure
we'd
thing.
be happy to invite you back.
Well, thank you.
So, does anyone else have any other questions? You can even unmute yourself and ask on your own if you
There's
like.
there's one more in the chat there.
Oh, okay. Um, can you speak more about the ethical side of collection? That is, how much seed to take from each plant versus how much to leave for wild propagation in that community?
As I think I touched on this lightly in that we only collect from populations that are large enough to really make a good collection from. You need more than 50 individuals. And of that population, we try and cap it at 20%. So if you have 100 plants present, you would only collect from 20 of them, tops. I think it's really easy to stay in that realm for the common species. And I would argue we are well below the 20% threshold the majority of the time. And I do think it really matters. I think at first, when I was really like hungry for seeds, I felt the burn of like, oh, but they're all here. I should collect more or something. And now I don't feel that. I think it is really important to approach seed collection from an abundance mindset, and both knowing that planet Earth really like has these plants, and we live in this incredible place that has so much public land that has so many plants on it. I don't think we need to be hitting populations hard at all. But the other part of the abundance mindset is to leave plants for insects, for the plant community. I love when I see our farmers just planting extra and leaving it for the birds, um, or the gophers in the cases of things that have taproots or bulbs or whatever. And I think it's just an important thing to also address the ethical issue of like taking seeds. I try now not to collect things unless I really feel like they're going to go somewhere. And after something's been sitting in the fridge for 3 or 4 years, I kind of am prone to tossing it in a mix to make sure that it goes out on the landscape before it loses its viability. And because we do this often, our practice just requires that we are constantly going out and collecting new lots. And I think this keeps us in touch with the earth and sort of, keeping our inventory going in and out in a way that really does, like, for me, it is geared towards abundance and reciprocity.
Great, thank you. And Merrick, thank you for that question. That was an important one. Anyone else? Or do you have any final comments, Vanessa?
Thanks for being here. Get out there and pick a few seeds out of planet Earth's garden, and yeah, leave some for the birds. Have your own experiments. Have a great time. It's great tinkering with seeds.
Great.
Good to see you, Vanessa, and thank you.
Good to see you, Gary. And thank you so much for hosting. You guys did a great job. Lovely host. Thank you so much.
Thank you. And I see a few thank yous in the chat as well. Well, thanks so much. We really appreciate it. And we'll look forward to Catherine's class on Saturday.
Oh, yeah. Go to the class. It'll be more fun than this for sure.
Okay. Well, thank you all for joining us, and please join us for our next classes and events coming up. Have a good night.
Farewell.